Hi, it's me again, Eliza Carrilbuguia, BSN A1 student, and I am here to report another lesson of biochemistry. So there are a few questions that needs to be answered, and I am here to answer all of those. So let's start. What is DNA? DNA is made up of chemical building blocks called nucleotides. Now these building blocks are made of three parts: a phosphate group, a sugar group, and one of four types of nitrogen bases. The four types of nitrogen bases found in nucleotides are adenine, thymine, guanine, and cytosine. Its compositions to polynucleotide coil around which carry genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. So these instructions are found inside every cell and are passed down from parents to their offspring. So one might ask, how does RNA differ from DNA? So let's talk about it. Before we differentiate DNA from RNA, let's discuss a few things about these two. DNA and RNA are composed of nucleotides that are linked to one another in a chain by chemical bonds called the ester bonds between the sugar base of one nucleotide and the phosphate group of the adjacent nucleotide. And both DNA and RNA are composed of sugar, phosphate, and base. So now let's focus on their differences. So as you can see in the diagram, DNA is a double-stranded molecule, while RNA is a single-stranded molecule. Next is their sugar. The DNA is composed of a deoxyribose sugar, while RNA is composed of a ribose sugar. Next is their nitrogenous bases. DNA has four nitrogenous bases, namely adenine, guanine, cytosine, and thymine. RNA also has four nitrogenous bases: adenine, guanine, cytosine, and instead of thymine, RNA has uracil. So right up next is their location. DNA is located in the nucleus, and some are located in the mitochondria, whereas RNA is present in the cytosol and ribosomes and rarely found in the nucleus. There is also a difference on their size. DNA is much larger than the RNA, with millions of nucleotides, while RNA only has hundreds. Also, DNA and RNA perform different functions in humans. So DNA is responsible for storing and transferring genetic information, while RNA directly codes for amino acids and acts as a messenger between DNA and ribosomes to make proteins. Furthermore, DNA is self-replicating, while RNA is synthesized from DNA. And let's all talk about those as we go on in this video. For now, let us answer this next question. What is the central dogma in molecular biology? The term central dogma is coined by Francis Crick in 1957. It basically explains how DNA makes RNA and RNA makes protein. It states that the pattern of information that occurs most frequently in our cells is from existing DNA to make new DNA or DNA replication, from DNA to make new RNA or the transcription, from RNA to make new proteins or the translation. And so the proteins here make phenotypes, and lastly the phenotypes makes us an individual. 3. What is genetic engineering? So genetic engineering is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. It involves the direct manipulation of one or more genes. Most often, a gene from another species is added to an organism's genome to give it a desired phenotype. In medicine, genetic engineering has been used to mass-produce insulin, human growth hormones, follastim, human albumin, monoclonal antibodies, anti-hemophilic factors, vaccines, and many other drugs. In research, also, organisms are genetically engineered to discover the functions of certain genes. What is DNA replication? Every cell in your body is the same exact DNA as the first cell. Now, how is this possible? It is because the DNA has the ability to replicate or copy itself. Now, isn't that amazing? This process is called DNA replication. Now let's discuss this process further. Since DNA is a double helix, it can be unzipped in the middle, and it will eventually make new strands on both sides. So now we have two strands of DNA. Basically, the enzymes involved in that process alone are the DNA helicase, which unwinds and separates double-stranded DNA as it moves along the DNA and forms the replication fork by breaking hydrogen bonds between nucleotide pairs in DNA, and then followed by DNA primase, which is a type of RNA polymerase that generates RNA primers. So primers are short RNA molecules that act as templates for the starting point of DNA replication. Then here comes the DNA polymerases, which synthesize new DNA molecule by adding nucleotides to leading and lagging DNA strands. The polymerase or DNA gyrase then unwinds and rewinds DNA strands to prevent the DNA from becoming tangled or supercoiled. Then a group of enzymes called exonucleases remove nucleotide bases from the end of a DNA chain. And with the help of DNA ligase, the DNA fragments join together by forming phosphodiester bonds between nucleotides. So basically in this stage, there are now two identical replicas of DNA from one original DNA molecule. 4. How is transcription and translation done by the cell? Transcription is the process of copying a segment of DNA into RNA. The segments of DNA transcribed into RNA molecules that can encode proteins are said to produce messenger RNA. First is this diagram. Involved in here is the DNA. And then there's also an enzyme called the RNA polymerase. And it will drive down the DNAs while it makes a copy of itself. So on that copy is going to be the messenger RNA. Then the RNA polymerase leaves eventually. So the DNA never leaves the nucleus. However, the RNA is free to leave and make proteins. And so that is how it works. Now let's move on. How is translation done by the cell? So the RNA moves out into the cytoplasm, and here it will feed through something called the ribosome. Every 3 letters of RNA will match on 3 letters of the tRNA, also known as transfer RNA. This is a different type of RNA. So the tRNA then brings in one amino acid that will eventually attach on the string of all the other amino acids in this protein. So from mRNA to a protein, and this protein here will make phenotype. And then, fun fact, the phenotype plays a significant role in every living organism. Dayon. So basically, translation is the process of translating the sequence of a messenger RNA molecule to a sequence of amino acids during protein synthesis in the cytoplasm. How does mutation happen? A mutation is a change in a DNA sequence. Mutations can result from DNA copying mistakes made during cell division, exposure to ionizing radiation. It could also be exposure to chemicals called mutagen, or infection by viruses. So let's name the different types of mutation or of how mutations really happen. So first is the germline mutations that occur in the gametes. Somatic mutations occur in other body cells. Chromosomal alterations are mutations that change chromosome structure. Point mutations change a single nucleotide. Lastly, frame shift mutations are additions or deletions of nucleotides that cause a shift in the reading frame. What is protein denaturation? Let us find out. Protein denaturation is the disruption of the secondary, tertiary, and quaternary structures of a protein molecule. It involves the breaking of many of the weak linkages or bonds within a protein molecule that are responsible for the highly ordered structure of the protein in its natural state. Now, there are agents or factors that cause denaturation. We have physical agents and we also have chemical agents. Now, in the physical agents, we have heat, the UV light, the ultrasound, the high pressure, and the violent shaking. And for the chemical agents that cause denaturation, we have organic solvents, acids, alkalis, urea, and various detergents. So now we are here in the last question. Here we are tasked to give 10 herbs that may be good for the lungs. So for the first one, we have ginseng. So ginseng is known for its potent antioxidant properties, which work well for fighting inflammation. Next is the osha root. So the osha root has an antihistamine properties which work well for soothing sinuses infections and other respiratory diseases. So next is the eucalyptus. It has been said that the leaves of the eucalyptus can be distilled and diluted into an essential oil, and this essential oil can be used for medicinal purposes such as treating sinuses, colds, fever, and other respiratory ailments as well. So next is the elderberry. The elderberry is known for its antimicrobial, antibacterial, and antifungal properties, which is so good in treating our lungs as well. Next is the astragalus. The astragalus decrease the chances of being infected by certain viruses and bacteria. Next is the horehound. The horehound is very useful for those with allergies, sinus infections, and other chest congestions. The bergamot as well has the ability to relieve feelings of stress and anxiety, and it contains properties that can make our breathing easier. It is antioxidant, anti-inflammatory, and antiseptic. So next is the lavender. Lavender contains antioxidant properties that work to combat the progression of the disease or the chest congestions and so on. So next is the peppermint. It is said that the leaves of a peppermint tree can be formulated into an essential oil, and this essential oil has been discovered that it has an analgesic and antiseptic and antibacterial properties that helps to combat viruses and other unwanted organisms. So lastly is the mullein. It is another herb that is traditionally used as a remedy for lungs and respiratory tract diseases or ailments. So first is the ginseng, the osha root. Next is the eucalyptus, the elderberry, the astragalus, the horehound, the bergamot, the lavender, the peppermint, and the mullein. So that's it for my report. Thank you so much for listening. Bye!